Okay, so thank you for inviting me. I'm a postdoc now at University of Vermont. I defended my thesis September 4th, so newly styled Dr. Sam. And I'm going to be talking today about living deepfakes. I never really thought about this work as deepfakes until you reached out in an email, but I really like this interpretation. So in the before times, before COVID, we would ask biologists, and also before we let the world know about living deepfakes, we would ask biologists to peer down a microscope with us. I'm going to slow this down. Hopefully that also helps. We'd ask biologists to peer down the microscope and tell us what organism they were looking at. We would tell them that this was in fresh water. They were 500 microns across approximately. They were moving in this way. What do you think this organism is? They would rattle off a list of Latin names, and to each one, we would say, no, I'm sorry, that's incorrect. Eventually, as you could imagine, they would get pretty frustrated with us. So we said, okay, well, we sequenced the genome. Would you like us to tell you that? Would that help you know and identify the species? Well, of course, you sequenced the genome. Why didn't you tell us that from the beginning? Okay, this is 100% frog DNA. But what you were just looking at were not frogs. They were not tadpoles. They were not like anything frog-like. How could that be? And this is deep fake number one, I think. So what we have here is a still image of 16 of those living deep fakes. They're kind of like beige blobs here on the screen. The floor of a petri dish was covered uniformly with red dye. So all the white streaks here are the behavioral history of these blobs as they were moving through, corkscrewing, and they seem to aggregate in the bottom right corner here. And they kind of spontaneously push these dye particles into a central pile. We actually don't even know why exactly they're doing that yet. We call these things computer-designed organisms, or CDOs. CDOs are the first organisms to evolve inside of a computer, to be AI generated. Instead of every other organism that evolved physically on Earth, modifying an existing body plan through mutations and recombination, these organisms were generated de novo and built from the ground up. The goal of my talk is to tell you in much more detail what these things are, how they're made, how AI is used to design them, hopefully how you can help contribute to make them better, what they can do right now, and why on earth we would want to do such a thing. So we'll start with the what. What are CDOs? Well, first, I think it's important to get a sense of the scale here. It's really difficult for people to conceptualize 500 microns. I have the slide in my hand. I'm not sure if you can see my webcam. It's not really important because you can also see it on the picture. But the reason why I want to show you this in my hand as well, whoops, is that this is not a deepfake image. These are not deepfake videos. These are real physical, well, in this case it's dead, but at one point living biological organisms. None of the videos or images that I'm going to show in this talk have been manipulated in any way. If they've been filtered, or if I'm showing a simulation, it will be very obvious, and I'll let you know. But this thing, this little tiny black speck in the middle of this slide, let's see if I can do this video. This is what it looks like zoomed in. Let me slow it down to real time. Now, I hope this video is coming across the stream. If not, I'll share a link where you can watch all the videos that we've released. But what you'll see here is a little blob of frog skin cells and heart cells. And the heart cells act as tiny little motors in this little CDO's legs. And it's pulsing and moving across the dish. So this was a novel organism that never existed before. For many, this kind of fell into the uncanny valley. This is kind of a grotesque looking, not quite totally real organism. We'll try to work on making them look a little prettier in the next round for you. So, okay, so we talked about kind of an imitation game in the beginning with the biologists, an uncanny valley now with zooming in at a particular scale. So I think in a lot of ways, this interpretation of these CDOs as deepfakes has really helped me expand my notion of what this work really is. So thank you. It's already been worth it for me. Okay, what are these things? We've covered that a little bit. Now let's talk about why we would ever want to make such things. Well, current technologies are made of metal and plastics, ceramics, electronics, chemicals. They degrade over time. They produce harmful ecological side effects. We have a really big plastic problem and all kinds of other pollution problems on our planet. We don't want to add to that problem. In fact, maybe we want technologies to go into the environment and clean it up for us. We don't want those technologies to then deposit more batteries and electronics into the wild. So the question is, can we make biodegradable machines? Machines out of something like frog cells that just, after 2 weeks in our case, disintegrate into dead skin cells. There's also jobs where traditional robots cannot go, but it would be really useful if they could. One example is inside of our bodies. Currently, there's no way to internally ablate a brain tumor, and we can't even see it until it's about millimeter scale on an MRI. If we could have some kind of little micro robots inside of us that are constantly monitoring our body, precisely delivering medicine directly to the point of disease, these would be really, obviously, extremely supportive of human life and amazing. The problem is that our bodies absolutely detest even the smallest amounts of metal. Even an organ that would save your life, if it comes from someone else, the immune system rejects it. So the question here would be, can we create biocompatible machines? I've talked about making these little organisms or living robots out of frog cells, but there's nothing really special about frogs. They could be made out of anything, any kind of cells, including a human patient's own cells, so that the immune system doesn't reject them. There's also the more purely intellectual pursuit of understanding what else is there. Nature has explored a very vanishingly small portion of the possible design space of living systems that could enrich and support human lives and the biosphere. We want to understand life really as it could be and how it could help us as a species. Okay, I hope I convinced you that we should be doing this. So now how are we making them? I should mention that this is work with Douglas Blackiston and Micheal Levin. They're both at Tufts University. They work in the Allen Discovery Center, which is a lab, a big center run by Micheal Levin at Tufts. And also Josh Bongard, who was my PhD advisor and now my boss as a postdoc, at the University of Vermont. This is our website, cdorgs.github.io. You can just Google cdorgs and it should come up. Okay, so here's another video, and let me try to slow it down. This is the build process. This is Doug building the xenobots. He starts with frog embryos. They're very, very early. There's no neurons in there. There's no specialized cells. He is removing part of that embryo, letting it disassociate in this medium. Actually, I'm going to pause this for a second. Can everyone see the videos okay? Is this coming across the screen? Or okay.
Yeah. Yes, it's working very well. Yeah.
Perfect. Okay. So then Doug is going to remove this extra outer layer of hard ectoderm. And what we're compiling right now is essentially a bunch of very small Lego bricks, sticky, living, moist Lego bricks that we're going to build a robot or an organism out of. So Doug is taking apart many of these embryos to get these particular kinds of cells. And he's also going to perform, I guess, what you might call some biological deep fakes to trick the cells into becoming different kinds of fates. They might have wanted to become skin cells, but he will trick them into becoming heart cells. Okay. Zoom. forward a tiny bit. Collect these cells and we place them into little dimples inside of a Petri dish. And we let them develop for a few days. And as they're developing, they are going to compact together and differentiate into some specialized kinds of cells and move about one another. And in the end, they form a kind of sphere. So this is hundreds of cells compacting and developing into a kind of spherical, tightly packed ball. And here's thousands of cells doing the same thing over the course maybe of 24 hours here. Okay. I just thought that was kind of a visually arresting video. I wanted to play it. And then here's what you get. So you get these spheres of cells, and this is like a hunk of clay through which Doug is going to sculpt the morphology of an organism. And what he's doing right now is he's using a very, very small micro cauterizer to it's like 13 microns across a wire that he's going to use to burn some of the cells away, and then he's going to scrape and burn them into the rough approximation of a shape. In this case, the shape is a quadruped. This is not a computer-generated blueprint, but rather was a human-generated blueprint. In fact, if I asked you to build a robot, or if we just look through the literature of soft robots, the most common robot is this kind of quadrupedal form. When humans design things, they tend to pick things that are familiar to them. So most robots look like humans or dogs or something else. So in this case, as a demonstration of the build method, one of the first robots, you'll notice that I throw those terms around interchangeably. I'm not even sure really what to call them. One of the first CDOs that we tested this build method on was this quadruped. I want to point out one other thing about how amazing Doug really is, besides the fact that this, as you'll recall, is a tiny little speck. Doug is doing this with one eye closed, essentially, no depth perception, because one of the lenses on the microscope has a camera in it. So the fact that he's able to sculpt the shape out is just remarkable. I think he told me an anecdote one time that he gets all these really incredible students at Tufts to help out, really bright, but if they don't have the right hand-eye coordination, they're not so good in the lab at helping at these tasks, as you could imagine. So he'll have some researchers that they're not really reliable, they don't show up on time, they play video games all day, but they have incredible hand-eye coordination because of that, and they're the best assistants in the lab. I always thought that was kind of funny. So you need to have you need to be like athletic to do this. I couldn't do this. So this is where Doug comes in. To give you a sense of kind of the complexity that could be possible at this scale, this is about the size of a tardigrade, which is this infamous animal that like can't be killed, survives starvation without air, in space, radiation, all kinds of crazy things. It also has pretty complex morphology, something like 8 legs, tiny little fingers. So yes, we're making pretty crude shapes, but who knows what kind of complex things we can make at this size. We're also not limited at this size, but I'll talk about that later. Okay, so now hopefully you have some idea of how CDOs are built physically. I want to give you an idea of where we see CDOs taxonomically fit into other synthetic biology. This is our best attempt here. So my understanding, to the best of my knowledge, synthetic biology before January 13th, 2020, when we released our paper, was all human designed. There are parts of synthetic biology that deals in parts of organisms called organoids. You might have cortical tissue organoid, a lung, heart, whatever. It's a little mini version of that organ in a dish. It can be really useful for kind of filtering and testing out drugs and just understanding developments. It's a really great scientific tool, but they weren't building whole organisms. As you can imagine, it's very difficult to design a whole organism from scratch by hand. And it's not something that they were really interested in. One thing that was of interest to this community was biohybrid designs. This would be, for example, something that is partially silicone. So in this case, it's silicone rubber. It's about 100 times larger than our CDOs, and they wanted something that swam. So they looked to nature, and what's a tiny little organism that's soft that swims? A jellyfish. So they designed their robot to be something familiar. It looks like a jellyfish. They implant heart muscle cells from a rat inside of this silicone scaffold, and they can excite it externally via electricity. So you can get this little jellyfish to swim. This is at the Wyss Institute at Harvard by some of the same people, I believe. A more recent work was another swimming organism. This one's about the size of like a penny. And they added some more bling to this one. It has a gold skeleton, and it uses optogenetics, which is a way to control cells' movement by light. Perhaps this also could be viewed through the lens of deepfakes. There's some really interesting literature on optogenetics and making mice do things that they otherwise wouldn't do when there's a little blue light shining on their brain. You can just look that up. But in this case, what we have are, again, a silicone scaffold shaped like a familiar object, a stingray. In the center is this gold skeleton. And on either side, there's little flippers that have hundreds of thousands of rat heart muscle cells embedded in them. They're optogenetically transfixed. So then when you flash a blue light on them, on the left side, all of the heart muscles are rowing on the left, and the stingray turns to the right. So this is an externally controllable biohybrid machine. It was hand designed. It was part synthetic material, part living material. Okay. What we introduced in this paper was kind of the left branch of this field, which are computer designed synthetic biological constructs. Now there could be biohybrids. We have not experimented with that yet, but what we did do was create an entire organism. And formally what we called the computer designed organisms that we created were reconfigurable organisms. It's kind of a mouthful, so it didn't really catch on. What did catch on was xenobots, which if you've heard of this work, that's probably the term you know of. It's just a cool word, so people latched onto it. Where that comes from is that the cells come from the Xenopus laevis frog. It's this really classical model system in biology, and that was used in Mike Levin's lab at Tufts, and Doug took those cells and repurposed them, reconfigured them into something else. Not a frog, but an organism with different kinds of novel shapes. There's also the sense that one of these organisms can be disassociated again and reconfigured into a new design. Reconfigurable has this really strong connotation in robotics with, I don't have to tell anyone at MIT, because MIT is the mecca of modular robotics and reconfigurable modular robotics. In our case, the modules are just really tiny living cells. Okay. Here's where the design comes in and fits the most closely with the class on deepfakes. We developed an automated design pipeline, and there's an input. The input to that pipeline are building blocks, passive building blocks. These are voxels, the 3D equivalent of a pixel. This is going to be in simulation, and contractile voxels. And there's one other input, and that's the desired behavior that you wish the output organisms to exhibit. In our case, we just want them to move into the right side of the screen. Here's what those building blocks look like in pairs. They're passive, two passive cyan blocks, and they're just sitting on the floor of the simulated Petri dish. And you can see the red changing to green contractile building blocks as they're contracting and expanding. And the problem here, the interesting design problem, in my opinion, is that we don't know how these contractile cells are going to sync in novel configurations. We know pretty well how heart cells are going to sync when they're in their default morphology, which is an adult frog heart, but we did not know, and we still don't know, how heart cells will sync in novel configurations. If you put a bunch of heart cells and skin cells in between them and you build a pyramid, Are they going to sink? Are they going to beat at their own pace? What are they going to do? The best analogy we've come up with so far is imagine that you're trying to design the shape of a rowboat. There are 100 people, it's a really large boat. There's 100 people in it, and everyone is rowing in their own direction, at their own frequency, wherever they want to row. Can you design the shape of that boat so that no matter how people are rowing, it goes straight? That's essentially the design problem that we're dealing with. We want to find body plans that cohere the unpredictable behavior of cells into more predictable whole body behavior that are going to work regardless of how these things are beating or regardless of how people are rowing in the boat. Here are some random configurations of these building blocks. And they don't do much. They kind of jiggle around. The red ones, again, are changing to green as they expand and contract. And the blue ones are sitting there providing passive support. Random combinations of these building blocks are not going to be great, but some will be slightly better than others. And by selecting those and making modified copies of those better ones, deleting the worst ones, rinsing and repeating, you get a process of evolution in silico in the simulation. After a while, with the proper constraints in place from the living systems, we get appropriate morphologies, the overall geometry of these bots, and the placement of the skin and heart muscle on them so that they move under random contractions. So these are the designs. Oh, well, those are the designs we might want to transfer, but let's talk about in more detail how we were getting those designs. So what we use are something called compositional pattern producing networks. These are very similar to GANs. They came out in 2007 by Ken Stanley. And what they do, I'd like to walk through just very simple ones, incrementally getting a little bit more complex so you understand how these networks work. The important part about these is that their input space is some coordinate frame that makes sense for the problem at hand. If you want to evolve a picture, then it might make sense to have the 2D coordinates of that picture be the input. And now imagine that the X coordinate was attached to an output that just painted an amount of green in this picture. So if X equals 0 on this side, it's white, and as X goes to, let's say, 1, it gets very green. Same thing happens when Y is connected to this very simple compositional pattern producing network, or CPPN. From Y equals 0 to Y equals 1, it gets progressively more green. You can add in some interesting activation functions in there so that it's all white up until a certain point, and then all of a sudden it's all green. You can compose these different activation functions together to get some really interesting patterns in space. And this website, I tried to go to it this morning, pickbreeder.org. It actually runs on JavaScript, so it wasn't working on my Mac, but it's from 2007, so we can give Ken a break. It was really cool. I'm sure there's a new version somewhere. I'll try to find it. It allowed users to start with a random CPPN and then pick which ones they want to breed. And then it would give you a dozen or so random mutations about this design. And you would pick just like you were artificially selecting flowers in your garden for breeding, but in this case in the computer, so very fast. So humans were selecting from this phenotype, this picture. What they really were doing was changing what this network was up here. So humans were selecting, and like I said, humans like to design things that look like familiar objects, so it's not surprising that they came up with something like a butterfly, although it is kind of surprising that this is possible with these simple networks. I made up these activation functions, but the point is that they're evolving the weights, the connections, how these different internal functions are squiggling about. And in this case, the output is red, green, and blue. And you can make something like a butterfly. Others have evolved these, artificially selected them, and under the scenes, the network is changing. There's no constraints for gradients. We're evolving it, so no assumptions there, or less assumptions. It can connect in any way you want. And they evolved a fox, which is kind of cool. something, fire wizard, whatever this is. You can also do this in 3D. And if you do it in 3D, it's the same thing. You just have a Z axis. And in our simple example of adding green, you're painting green in this cube. Okay. So in 2013, Nick Cheney and his colleagues, including Hod Lipson, applied these to robots. And that's the same encoding that we use in our computer designed organisms. So the input space is something to do with their 3D spatial coordinates. At some resolution, you can pick it, you can upscale it later. They're indefinitely scalable, whatever resolution you pick to query the space in, that's what the network will paint. So here's some random network, let's say. It has different activation functions, different connections. The important part are the inputs and the outputs. So the inputs are the XYZ, and the outputs are going to determine whether or not there's material there and what kind of cell type it is. So for example, we could query this network at y equals 1, x equals 1, and z equals 0. If there's any art majors, I apologize for the perspective on this. And let's say that the network says yes. At x equals 1, y equals 1, z equals 0, there is a cell there, yes, and its material is passive. So places a passive block in that position. Query the network again at the next coordinate, and it says no. At 1, 2, 0, there is not any material there, move along. And we do, and we query the whole space, and it tells us where to place contractile and passive voxels. And because the input space is the physical coordinates, the output is correlated in space. So that gives us some nice regular patterns that are good candidates for transferring AI designs to physical artifacts like robots or computer designed organisms. So this was one of the evolved robots that was produced by this generative network. Here's 100 of them. So we restarted this process 100 times with different random initial conditions, and each one of these is the best one after 1,000 generations evolving. Each one had a population size of like 50 robots, designs, and at the end we took the most fit one. Some of these we can build and some of these we can't. So this is something interesting when you're trying to transfer generative models to a physical artifact. Some of them are just unbuildable. And some of them you can build, but they're not going to have the same behavior in simulation and reality. So there's extra complications to getting these kinds of generative things into reality. As just one example of the kinds of constraints we're dealing with, we found that with our current build method, there's a limitation on how small a gap can be in the body that will persist over development. It was about 100 microns, which translated into our model at this particular resolution to about a gap of 1 voxel wide. So any designs that have that gap, we just filtered them out. So they're out of consideration. There's also ones that we weren't interested in building. For example, ones that are all red, they're all heart muscle. But we thought that designs that have more passive tissue are more interesting for us because in the future, we want to scale these to not just being skin and heart muscle, but to having other kinds of interesting organs like sensors and stomachs and whatever else. And we thought that it would probably be easier to place that in the passive and the cyan tissue. So there are some of these constraints. I have a slide at the end of the deck if people are interested in the details. But we set up these constraints in an automated way so the system would just spit out some of the very performant designs that adhered to our constraints. And here is what it came up with. And for the moment, Doug is better than any bioprinter at creating some of these. So this is one of these mostly passive, as you can see from the cyan, highly performant designs. And here's Doug's recapitulation out of frog cells. It's kind of like an L or gamma or something. So why is Doug better than a bioprinter? Well, first of all, they're very, very small. So you need to have a bioprinter of very high, fine resolution. Not just that, unlike 3D printers that print a toy, living cells have their own agenda. They don't stay put. So when you're printing them, you'd have to constantly be nudging them and scraping them and forcing them into some position, or else pushing their developmental trajectories in the right way. That's somewhere we'd like to go, but at the moment right now, these are all manually built. Okay, so without further ado, here's some more of Doug's creations from the AI generated designs, a kind of little pyramid, a biped, a different topology for the mathematicians, a torus. To get a sense of how well Doug is really placing the different tissue types, however, we can use techniques to kind of make the different cell types glow different colors under different fluorescent lights. So we can shine certain lights on them and make the skin cells glow green, ectoderm, and the heart muscle glow red, the cardiac cells. Okay, and we want to see the most important thing, not that we can just transfer the structure, but that we can transfer the behavior. Because we're not making statues, we're making organisms. Organisms behave. So this is some visual evidence, and I have later slides to prove, if anyone wants to be a stickler, that we do have p-values here and repeated trials for proving that the design transferred from simulation to reality. You can see in silico or in simulation, it's moving at a perspective, but in vivo, we're looking down a microscope. It's hard to get the perspective. And these are walking organisms. So they are in water, but they're pushing off the bottom of the dish. This was something that was easier to prove transferability than the swimming ones. To prove that this is due to the design. It's not just that we stick together any cells and they move, but that we need a smart system to generate good designs. We flip the organism onto its back. So this changes the geometry and the distribution of tissue, and that inverted organism no longer moves. So it is due to the design. All of them don't just move. This is an important control experiment. Sometimes it's difficult to even think about what these control experiments could even be. You might also notice that this organism looks even kind of grosser than the other ones. Doug can make them at varying levels of accuracy. And when forced to make repeated trials, he did kind of a more quick and dirty method of producing these, which still recapitulates the design. There's big hind legs, short front legs, muscle here, but they're not shaped as beautifully as in this still image. But what's interesting is that doesn't matter. We're transferring behavior. Right? We don't care about really what they look like. We care about being able to predict in simulation how they'll behave and then building them and realizing that behavior. Okay, so there are some applications that we found here. One of them was that, I'm not sure if you can really call this an application, but we did. It can automatically self-repair. This is something that's very difficult to do in traditional robots made out of artificial materials. It's kind of, there are a lot of soft robots that are trying to figure out ways in which they can be punctured and heal very small wounds. But one of the things you get from building out of cells is this ability to self-heal for free. So cells will do this automatically. When we damage the organism, it automatically zippers itself back up. So now the organism has a kind of harder ectoderm. It's different than that minimal concavity size during development that would be closed. Now it's kind of formed the shape it wants to be. And when we damage it, lacerate it mechanically, it will seal up that wound. Again, we're not actually even sure how it's doing this. We're trying to investigate what's going on. What we do know is that we can view these cells, and cells are themselves very complicated, complex machines that sense their environment. They talk to their neighbors. There's about 5,000 cells in this little gross blob here. And when we do cut it, all the cells yell. They flash up. They flash lights. They're passing ions to each other. We can see them communicating electrically and chemically. They're also probably feel it mechanically that some of their neighbors are in a different position. So we know that they can feel the wound. We're not sure if they're saying, hey, you know, we need to heal ourselves, or if this is basically like a zippering up that's automatic and mechanical. It's something we're investigating, but it's an interesting property of these systems that they can self-heal. We also investigated some other applications like object manipulation, in this case, block pushing. So we evolved designs to push a block. We didn't prove transferability here, but we did see similar behaviors in reality. You can see this little guy come in from the side of the screen and start pushing a pellet in its dish. Here it comes. So we thought that was kind of cool to see in both simulation and reality. In simulation, it was nice because we saw different morphologies evolve from the different kinds of behavioral goals that were input into the pipeline. So this one evolved a little kind of end effector to hold the block, a tiny little hand. So that shows that when you have different inputs, you can get different kinds of designs that are suited for those kinds of goals with the given building blocks. We also can place multiple organisms into the same dish. Slow this down for you. And we can see that they kind of pair up. They do a little like barnyard dance. They like attach to each other and swing about the combined center of mass. We also saw something similar in simulation. I'll admit it's going to be difficult to see, but it's going to happen in like 2 seconds. Two simulated designs are going to link up mechanically and spin about each other. So it was cool to see some of these behaviors be predicted in simulation and in reality. Some of the behaviors in reality being predicted in simulation. But again, we didn't prove any transferability on these collective behavior tasks yet. The fourth application was something that wasn't even designed beforehand by humans. We got one of these designs and it had a hole through the center, and Doug built it. And we thought, well, there's a hole in that. We can put something in it. So we exapted that design. We exploited it for a different kind of adaptation. In this case, object transport, or some will see this as the rudiments of intelligent drug delivery. 1 second, here we go. Place a yellow pill inside, and then we're gonna zoom out, and you can see this little organism carrying the pill across the dish. What's particularly interesting about this is then the AI produced a design that inspired us to change the kinds of tasks that we were even asking the AI to produce. It wasn't just producing diverse possibilities and designs for a particular goal that we imagined. It changed the space of things we were even imagining that the system could do. So then we actually explicitly evolved designs to carry a little pill inside of a pouch in the body. And this is where I think that generative models and deepfakes can be a really great tool for roboticists, whether they're building out of cells or synthetic materials or synthetic biologists, to help them kind of think outside of the box. And that's why I am really excited about this space moving forward. And I hope that there are people in this class that are interested in this work and would like to help out and improve our very rudimentary compositional pattern producing networks and help us design even better computer-designed organisms, xenobots, in the future. And that is all I have. I would love to take any questions you might have.
Wow, this is amazing. Thank you so much.
Thank you.
Yeah. Yep. A lot of people are commenting in the chat. Anyone have any question? I had a lot of question, but I'll save it if anyone else have it before. Oh, Patty has a question.
Okay.
Yeah, one question is, do you improve the simulator based on the observations of the behavior in vivo, and especially if it's different from what was predicted in the simulator?
Yeah, that's a great question. Oh, sorry.
Not mine.
Actually, a recurring organism isn't behaving. Luna. So I was ready for this question, so thank you for asking. Just make it so I can see everyone's video at the same time. Okay, so We initially have an input to our design pipeline. And these are the types of tissues and the behavioral goal you'd like it to exhibit. We simulate something, we send designs to reality, and it outputs an organism. The first time we did this, we got, the first time we did this, we got organisms that galloped like horses across the simulation. As you can imagine, this would be very difficult to make out of living materials. So we had to go back into the model and, sorry, Luna, come here. So we had to go back into the model and say, look, one thing we're noticing about these designs now is that they are hopping off of the bottom of the dish. They're airborne for half of their gait cycle. That's not something that we think we can produce with the current power of these heart muscles at this scale. So a little line was just added to this design pipeline that feeds back into the simulation constraints. Those constraints were manually designed. And it's something we'd like to do in the future is figure out how to automate this process a little bit better. But we constrain the simulation now to simulate these hydrodynamic drag forces, and we randomize the actuation to the kinds of unpredictable behavior that we saw on the slideshow. So previously, we were allowing really coordinated muscle movements, which resulted in this jumping. We randomized that, so that reduced the muscle power and the coordination. And on the second pass of the pipeline now with these constraints, we have behavior that better match the real dynamics, and we can measure this quantitatively. They keep part of their bodies in contact with the dish at all times. We know this happens in reality because the organisms have negative buoyancy. So this was a really nice target for us to quantitatively bring simulation closer to reality. We think the future work would be to encode expected cell coordination, not just figure out bodies that are kind of denoising the random, the unpredictable. Can we actually predict if I have heart cells and I put them into a pyramid, the ones on the left corner will sync up with the right corner or not? What's the smallest, if there's a dumbbell shape, it's really small in the center, will the sphere on the right sync up with the sphere on the left? We don't know yet, but we think that we can start to perhaps predict that in the future. I hope that made sense with all the barking and the lawn mowing going on.
Yes, thank you. I had one other very quick question. Do these cells reproduce?
So right now,
Divide. Yeah.
Right now, these organisms don't have any means of reproduction. They're just skin cells and heart cells. After about 14 days, they disintegrate into nothing, so they're not reproducing. There's always the possibility of bioengineering, self-reproducing ones. Obviously, that would come with a lot of ethical concerns. Yes, go ahead.
Although the question isn't whether the entire little mini organism reproduces, but more each of these individual cells.
Got it. So
Yeah.
the cells are not dividing during this developmental process. They are differentiating and compacting together. So there's a fixed number of cells that, because of when they're taken from the embryo, yeah, they're not reproducing themselves at the moment. And I think we'd like to keep it that way.
That's really cool. We have a question from Sasan. You want to unmute yourself and ask the question?
Yes, I'm sorry to ask you a question. I was wondering what kind of tools you use for this kind of amazing simulations. What types of softwares and
Thank you.
Python packages maybe.
So do you have a website? Created a new simulator. It's GPU accelerated. It wasn't used in this paper, but all of our work now uses it. It's the same idea. It's based off of voxels. If you type in Voxcraft, kind of like Minecraft, but with Vox, .github.io or just Google it. You'll find this whole kit for making soft robots, but on the design page, it's agnostic to what you're building these voxels out of in reality. And we have a simulator and a visualizer, and there's a Google Colab notebook to get started. And you can put these little...
Hi, everyone. I'm going to show you how to use...
you can put these in different backgrounds and other things. We are using an evolutionary algorithm that I don't know if it's in this code or not, but in the paper and on CDOs, we have everything here. This is kind of our home base for videos. Feel free to use them in whatever you want. They're all Creative Commons license, non-commercial, or you can use them for whatever you want, but the code is non-commercial use. And yeah, so the code that we used was Python, but the simulator is C++. And if you want the more up-to-date GPU accelerated CUDA simulator, go to Voxcraft.
Thank you so much.
Yep.
Yeah, and Olivia, I think you raised your hand.
Yeah. Thank you so much for the presentation, Sam. That was really mind-blowing. I have 2 questions. First one is about whether you can use the results from the simulations you've done in the design space, or you have already a design space that has these basically parameters you can explore to get the organism or whatever it's called to achieve a certain objective.
So I missed the noun. Did you say line speed?
Design space.
Oh, design space.
Yeah.
Sorry, could you repeat the question? How did we figure out the design?
Yeah, correct me if I'm wrong, but it seems like you, the project started out creating random like kinds of arrangements of cells, and then you chose the best ones. But then moving backward, could you use the simulations to create a design space that you can now optimize over and then get the best possible design from the design space?
Got it. So the way that these designs are being generated is with this compositional pattern producing network. You are wondering if we can somehow input data directly into this generator from the real world, from like videos
that you've gotten from the simulation.
I think that would be that would be awesome. Let me see if I have a slide about that. Go. We have this, this pipeline. What we proposed was a pipeline. And in this pipeline, the moment we're producing all of these designs and they're getting sent to reality to be built, and then constraints are coming back, it would be fantastic if part of this one arrow labeled G coming back from the real world contained data from reality quantitatively, automatically adjusting the simulation. At the moment, no. What we're doing, actually this wasn't the figure I wanted to show. I wanted to show, bear with me one moment. There is kind of a cartoon of the pipeline. And we basically did the simplest possible thing. We generated all of these designs in probably an inefficient manner, and then we filtered through them to find the ones that would pass to reality. There's certainly more efficient ways that you could do this, and you could incorporate data directly into the design process. Second. Aha, okay. Promise it was worth it to get to this. So there is building blocks on a goal come into this. We have evolved designs. And then what we're doing is we're taking all these evolved designs and applying like a filter to them. But that filter could have been built into the design process itself. This was just the easiest way for us to do it right now. To prove transferability in the first case, it turns out it's easy enough to just evolve a diversity of forms, sift through them, find a robust design that is robust to a whole sequence of random possible contraction patterns, and then build it. We call it a generator, which is basically just Doug sitting there building robots. That could be automated too. So there's lots of parts here that are manual that we'd like to make automated, including, as you mentioned, the constraints back from reality.
Great. Thank you so much. The second question is a bit more whimsical. I was wondering, it seems like part of the constraint is how small everything is. So I was wondering if it's possible to make a giant blob of cells as a voxel and basically upscale everything, and then you are able to observe the organism in a bigger sense, or does the hard cell not allow for that kind of scale?
Yeah, so we can scale the organism, we can scale the simulation, definitely, which is one of the really nice properties about these central pattern producing networks, is that they're scale invariant. So in simulation, we can scale them to whatever resolution we want. Although even drawing this thing took a long time in Matplotlib. I don't recommend trying to do that. It actually drew like every single voxel, even the ones that are in the back. In reality, there are some interesting constraints. So if you want to make a really big xenobot that's not just like this tiny little speck, but that is the size of like a baseball or something, or a soccer ball, well, you need a lot more cells. It would take a long time, and you might need bones for support. So right now at this very small scale, skin and heart muscle are enough to support themselves. The design problems get interesting, and they're different at different scales. There's also a problem in organoid research, which was these synthetic biologists that are designing these little tiny miniature organs. If they want them to be big, there's this like kind of bound on how big you can grow something without vascular networks in them. The center will tend to die if it's not getting like blood flow through it. I think that this poses an interesting question. We could probably design geometries that don't need a vascular system that could be bigger. So there's all kinds of really exciting non-intuitive design problems that things like generative networks could help us realize.
This is really fascinating. Thank you so much.
Thank you.
Yeah, and I think at MIT there's also a class in the biomaker space called How to Make a Biobot, and this was by our friend Ritu Raman, who made the kind of a muscle robot that just walk around. I think they did not use AI to generate the form. It's very simple, but if anyone want to get a hands-on experience on kind of being dark and making this bot, that class is really fascinating. Yeah.
Ritu Raman is awesome. Her work is a huge inspiration of mine. And I've been trying to convince her to apply AI to her designs a little bit on Twitter, but we'll see if she listens.
Yeah, we can definitely connect you with her. She's our friend. Yeah. Any other question? One thing that I think is very funny when I saw your presentation in the beginning is you showed the genome of this cell, and they're actually like a natural genome. You actually did not genetic engineer them. And it's kind of funny because, people, when they see this, they might think that, oh, this is like a crazy bioengineering project, but it's actually much more natural than probably the corn that you eat. It has been kind of engineered many times.
Yeah, so this is also different than a lot of synthetic biology, which changes the genome. So these are GMO-free, organic biobots that you can probably buy at Whole Foods for $100 or something in the future. This is kind of expanding our notion of the plasticity that's inherent in development. You can't look at a sequence of DNA and predict the shape that's going to come out. There's a lot more plasticity in life than that. And the questions are, can we push that plasticity into new kinds of forms and functions that are useful? And what can they even become? I think is really interesting questions.
Really cool.
Sam, may I ask you a question?
Sure.
I think, well, first of all, also thank you so much for your presentation because it's really incredible and very inspiring. I was looking at your work and you were saying how you started to see tendencies of these xenobots collaborating. And I'm wondering if you've considered how your pipeline might be able to scale if we think about something like swarm robotics. So if you had like many of these things operating together, do you think that you could actually simulate like a crowd behavior of these bots?
Yeah, so one of the things now that's possible that wasn't possible in our first paper is to even simulate swarms multiple times. So we did simulate some swarms and I showed a video of it in the first paper, but excuse me, that took a very long time to simulate and we could not evolve swarms. We could not predict the crowd behavior and then transfer it to reality. But now with our GPU simulator, which you can check out at voxcraft.github.io, we can simulate all of the voxels in all of the robots in parallel. So this allows us to simulate large swarms. We could simulate this entire 100 robots at once. Some of the things that I think there's a lot of stuff that could be taken from the swarm robotics literature and applied to this. It'll be interesting to see how the living systems kind of can aid that process. So you might not need to simulate everything perfectly, whereas in a swarm robotics system made out of metals and electronics, you might need to precisely figure out where all of these robots are going to join together and like work together in a certain way. But in a living system, I mean, they do things automatically. They are automatically doing this. So if you can get them to do what you want them to do, which is challenging, then there's all of this aided stuff that life kind of gives you. But the challenge is that living systems kind of resist forms and functions that are imposed on them. If you can break that barrier somehow, if you can learn to predict what they'll do under certain situations, then you get a lot of benefits like the self-healing and the spontaneous collective behavior and other things. So yeah, I think that's definitely where we want to go. There's lots of applications where swarms would be a great fit. A single tiny speck of a robot wouldn't be very useful in many applications in the real world, but a whole swarm could maybe, you know, group together microplastics in the ocean or something like that and then just disintegrate away. So yes, swarms are on our agenda for sure.
That's so cool. And in terms of trying to conduct their behavior, do you imagine that they would have to communicate with each other, or is it like this external source, like with the what was it called? Optogenetic?
We want to explore everything. What we're trying to figure out now is, are they communicating? So we're coming out with a couple more papers on this. We didn't even know this was possible 2 years ago. I was showing some like designs that just looked like, you know, the voxels to Doug, and then one day he just built the thing out of cells, and it kind of changed everything that we were thinking about. This is not something that I knew was going to be possible like anytime soon, and not something I thought I would work on. Doug came around and built it, and it changed kind of the game. Sorry, I forgot the question.
That's really cool. The question was about communication and whether you think that the way to go would be that they'd have to somehow communicate with each other, or if there'd be some kind of centralized control that's shone down upon all of them.
Yeah, the light from on high. So we didn't know this was possible. Now we're exploring everything. One thing that we see now is we can view the like bioelectricity of these cells. So all cells communicate chemically, electrically, mechanically, whatever. What I think some people find that they didn't know was that cells communicate electrically, not just neurons, all cells in your body. We can view those electrical message passing in each individual xenobot, CDO, whatever you want to call them. We can also see them, we can see flashes go across organisms. We don't know if there's any informational content in there that's being utilized by these bots, and if that's even affecting how they're moving, or if they just tend to kind of make piles like this just because of their wind-up toys. They're wind-up toys made out of heart muscle that move in a certain way, and that will get you a pile. I would ask the question, is there any difference? Like is there any real significant difference between that, whether it's electrical or mechanical? If they're making the piles and you want piles, then you're done. It doesn't matter how they do it. You don't need to predict everything in simulation. But yeah, I think the questions for the biologists about the communication are really at the top of their agenda as well.
That's amazing. I know that there are so many questions now that you can ask.
I have a question. I don't know if you can hear me well, but you mentioned having different kind of cells beside the moving voxels, like maybe there's a sensor cell. Now, I was wondering how the arrangement of these cells to make this like large organism could help in different type of voxels besides moving voxels. Like what other functions or goals can you have?
Yeah, so what are the limits of an organism without eyes? Well, it can't change its behavior based off of distal perception, things that are going to happen in the future. It can't do that in an intelligent way. It can't follow gradients very well. We don't actually even know, because again, each cell does sense things, and the cells themselves are really complex machines. So the benefit of like major sense organs like noses, eyes, and everything else, we don't yet know what those will be, but it's something that we're definitely interested in doing and making these more competent. What's interesting to me, I think, is that even these really simple ones can have applications. So what can you do with something that just moves in circular patterns, that's biodegradable, biocompatible? Well, it could swim around in your arteries and scrape out plaque. Doesn't need to have eyes for that, just kind of scrape. It could scrape away calcium deposits in arthritic knee joints. I think there are applications in the kind of like dumb CDO space. But then there's also going to be things that you want to do that are probably going to require more sophisticated perception, and how we're going to simulate that and predict it in our models is going to be very interesting to try to tackle. I have no idea how we would do that at this point in time.
Thank you for the great lecture.
Thank you.
One question from Janet. Do you want to say it? Janet?
Yeah. Hi. Yeah, the question is, have you tried exposing these cells to different biologically relevant chemical gradients? For example, calcium or various other, potassium and so forth, that these cells would normally be either excited by, but certainly affected by?
In the first paper, no, but in our work now and the whole suite of systems and different kinds of cells that we're exploring now, yes, we are exposing, the group at Tufts is exposing them to different kinds of chemicals and seeing if they can follow these gradients and what they do, and if they'll eat and other kinds of interesting questions. It's not work that's being done in my lab, so I can't really speak to the details about it, and it's not yet published. But I can say that from the design perspective, it's going to pose some really challenging and interesting design problems for the in silico design. Where
I
do you
can imagine.
Well, let's say, just kind of going off of the last question as well, if you can have a sensor, where should it be placed on the organism? Should it be on the top or the bottom or everywhere? So these will be interesting things that also will probably require more finesse in how the constraints are feeding back into the simulator. There's a lot of work to be done. Basically, we did the simplest possible thing that we could do to prove that we can transfer designs from simulation.
Right.
Yeah, and next we'll try all of these crazy, cooler, more sophisticated things. But yes, the gradients are coming, I can promise you that.
Okay, I mean, it's clearly asking for trouble in terms of the simulations, but
yeah.
Yeah, I think another interesting idea, tying back to the idea of deepfake, was that maybe instead of kind of training these model to learn the physics of the cell from the simulation, maybe you can kind of create a model that go and sort of like mimic the tissue of existing living organism, and then kind of translate that into generating this cell. So kind of deepfaking the composition from existing organism which evolved for millions of years. That could be an interesting idea to explore too, I think. Yeah.
Yeah, I think that's exactly how one might go about trying to predict the coordination of these tissues in novel arrangements. We'd have to be careful about introducing biases into this. We want to get different novel arrangements that we don't necessarily see in nature. But we do want to improve the complexity of the design. So yeah, you carefully pick and choose your biases and to make some progress. And I think that is definitely, I think you've targeted the area where I think the kind of GAN models can help this work. And I would love to collaborate with anyone that's interested in applying those to computer designed organisms.
That's really cool. Any last question? Great. I think people are very excited. I think they still need to think about, what they're gonna do. And I think I'm really grateful and really appreciate that you come and speak in our class, even though it kind of is a little bit different than the rest of the thing that we cover. But it really brings kind of new and fresh perspective, and hopefully some of the algorithm that people have been kind of playing with and learning from auto class, like we were talking about pick-to-pix algorithm that do like pixel translation. Maybe that could be applied to cellular configuration. Who knows? But it's really cool to have you. And as I said, I'm a big fan. I'm a biologist myself before I kind of start doing more human computer interaction research. So I'm really grateful. Yeah, thank you so much. So everyone, please give another round.
Thank you.
Yeah.
Thank you for inviting me.
Yay. Patty, any kind of departing note or thought?
Thank you. Thank you so much. Super interesting work. So
awesome.
Thank you, Patty.
Yay. And yeah, so as a reminder, I have some kind of closing slide. We see these amazing projects that are related to generative algorithm and generative AI. The lecture, yep, sorry.
We don't see slides.
Oh, sorry. Okay. Yeah, and the class lecture will be posted on the website, deepfakes.media.mit.edu. So if you wanna, rewatch any of the class lecture, feel free to do that. Again, homework 0 is posted on the platform, so go and download the Colab and play with it. And we have more homework coming up soon. We are a little bit behind, but we'll catch up very soon. And if people have problem tomorrow, I will have a virtual office hour, 1 to 2 p.m. If anyone want to come in and chat, we're happy to do that. Some of the students already kind of pitched the final project that they want to do. So you can also come in and talk to us about that as well. So that's pretty much for week 5. And we are very excited. Thank you.
thank you, Pat.
Okay. Great. Thank you so much. Bye.